Okay.
All right, I think I am unstuck here in terms of advancing slides, so we'll try this again. Okay, so this is the same workflow slide I showed earlier. So now what we're going to talk about now is how to put this together into a what are the specific questions you need to answer if you're actually wanting to take a sample set and a particular experiment through one of these workflows. So most of the questions have to do with, number one, how do you prep the samples to get them ready to go into this first trypsin digestion step? And that, that depends on what kind of samples they are. So we'll talk in a little more detail about that in a minute. And then depending on which mass spec experimental workflow you use, there are a lot of variables here that can be tuned, that can be adjusted to fit experimental needs. So for example, this offline fractionation can be easily manipulated to collect more or fewer fractions and therefore use more or less instrument time depending on what you need. So ideally, you want to use as much instrument time as you need to use to detect as many the number of proteins you want to be able to detect, but you don't want to use more because more instrument time costs more money, and so we don't want to spend a lot of extra time collecting data that ultimately ends up not being useful to answer the biological question. And there are other other parameters in these workflows that are where similar considerations need to be addressed. So we'll look at some specific questions that need to be answered in most cases before you do a proteomics experiment. So the first is, what question do you want to answer? What's the hypothesis? And we touched on this before. You don't necessarily have to already know the answer. You don't have to know. Your hypothesis doesn't have to be, protein X increases threefold under this biological condition. So your hypothesis may be as as vague, I guess you would say, as something biologically interesting happens under these conditions, and so I want to look at I think this is related to changes in the proteome, so I want to start trying to figure out what those are. That's a perfectly good hypothesis. Some would say fishing expedition, but Rick will tell you that it's not fishing, it's catching, if you do it the right way. But in some cases there are specific questions more specific questions that you're interested in answering, and it's important to address those up front to make sure that that's incorporated into the experimental design to the extent it can be. So another common question, very important question, is how many replicates do you need to incorporate into the experimental design? And the answer is, if you ask a bioinformaticist, the answer is more. So that's 5 is the new 3 and 7 is going to be the new 5. But the absolute minimum for any type of real statistical and quantitative analysis is 3. So you always want to have minimum 3 biological replicates. You can still do pilot type analyses with single or dual replicates if you need to, but it's not recommended, and it generally doesn't yield standalone publishable data without at least 3 replicates. Now, depending on the type of sample and the type of experiment, if you can get to 5 replicates, that's generally much better, and it puts you on a sounder footing in terms of data analysis. But the trend here so far is to have a conversation about all of this stuff before anything happens. So one of the first thing we like to do when we talk to somebody who we've not worked with before is to schedule. We used to do conference calls or something. So now it's all on Zoom. We want to have a meeting with the biologists, the people who are going to do the work in the lab, the mass spectrometrists, and the bioinformaticists. So we everything to the extent it can be is designed up front. Any questions that you're going to have, anything you want to get out of the data, you want to make sure the people who are going to analyze the data know about that before it gets all the way to that point. And it turns out that, well, we really needed 5 replicates to answer this question satisfactorily, and we just spent a lot of time and money on a 3 replicate design. So you want to avoid that kind of thing. You know, other questions are, you know, things like you know, post translational modifications. I think I have a slide on that here in a minute. If you know, if you're looking for changes in phosphorylation, you need to let the proteomics people know that before all the data are generated, because we do the experiment differently if we know that you're looking for something like that. So definitely, you know, this is usually this is, you know, proteomics analysis is not the type of situation where you want everyone to be blind to whether it's you know, double blind type experiments don't usually work as well this way. There, you know, the exception I guess would be with certain clinical samples. You would not necessarily have all the patient, the group information there. But, okay, so yeah, as I was, you know, this is, you know, what I was just saying. If you know, if you're interested in acetylation, you know, that needs to be. Don't wait until you get the data set at the end to ask if there was any acetylation detected, because it'll be too late at that point to salvage the experiment probably. All right, so I'll pause for a few seconds here and see if anyone wants to jump in or has questions, or if there's something you want to talk about along the lines of what we've been covering here so far.
So let me just say, this would be a great time if you have your project that you're just dying to ask a few questions about it. Just go ahead and be that guinea pig so everybody else can kind of see how this process goes.
So I have this salt-inducible kinase that we've already shown to be involved in sleep as well as fat storage. However, we're curious if it changes phosphorylation partners when the nematode's sleeping versus awake. So I've been trying to figure out the best way to like phrase the question, or as well as like the type of workflow we do at the Proteomics Center we have here. Do you think you could help with that?
Yeah, that sounds like you know, something we could definitely design an experiment around. So there are a couple of different ways you can approach that. So one would be you know, just a global phosphoproteomics experiment, which we would, for, you know, we would use the tandem mass tag platform for that. So you would have a, you know, control versus you know, a knockout or a, you know, inhibited kinase treatment, and then you just, you know, grow up cells or whatever it is, or C. elegans organisms. I think that's what you said. And then we just apply the TMT platform and add a phosphopeptide enrichment and look broadly across the entire phosphoproteome and see what changes. So then the other way you could approach that is if, you know, if you're looking for, you know, what proteins does your kinase specifically interact with in the cell, you could then, if you've got an antibody or a tagged version of the kinase, you can pull it down and then analyze the protein complexes that you isolate to see what the differences are in interaction partners with that kinase. So probably the pull down experiment, we wouldn't specifically look for the phosphorylation sites. We would just look for what changes in terms of protein interactions. And we would probably do that by data independent acquisition because that's more tolerant of, you know, usually with pull down type experiments, you're limited in terms of the amount of sample you can isolate. So you generally don't have enough for isotopic labeling.
Okay, that seems like a better option considering like with nematodes, since they're barely a millimeter in length, getting a lot of protein content is kind of laborious in itself. Thank you.
Okay. Great, perfect. Anybody else?
Okay, I'll jump in there. So I have some various different cells from different patients of a particular disease, and the disease is manifest in a tissue that has a lot of tension in it. So mechanical tension. And what I find is that these cells, if I age them in vitro, they tend to more robustly be respond to the mechanical tension of the tissue. So what I want to do is I want to compare the response of these cells in an unaged condition versus an aged condition in the presence of the mechanical tension. And so in order to get them in this model, they have to be encased within an extracellular matrix. And so, a number of questions I would have is like, what's the minimum number of cells I would need in the different treatments, and how to prepare the tissue or the cells, like I mentioned earlier. Sometimes I just collagenase the tissue and spin down the cells. But, where would I go from that point?
Okay, so we can skip ahead a slide or two here. So there's a, I think I've got a, how much do you need type question here. So yeah, so for here we go. So it depends on the technique. So for TMT, we need, the labeling reaction ideally requires, 50 to 100 micrograms, in that range of total protein. So if you can, that with most cells, that's somewhere in the neighborhood of, 3 to 5 million, something like that, depending on the size of the cell. So that can be, it's, in cell culture, that's usually easy, but, with your setup, that sounds like it might be more difficult. So with the data independent approach, because we don't have to label and we don't have to fractionate, is more forgiving in that regard. So we can get away with, we'd still like to have, 10 micrograms total protein per sample to get a good reproducible trypsin digestion across the sample set. But that, obviously that requires a lot fewer cells. So does that sound like it's, in the vicinity of reasonableness for what you need to do?
Sure, yeah, it'd be a lot of replicates, but I can handle it.
Okay. Yeah, we should, it sounds like, with yours, that's going to be, we should probably talk offline about the best, how to get to figure out how to get your cells out of the matrix and do the isolation.
Okay, great, thank you.
Okay, so I'll go ahead and go through a couple more of these slides here, and then we'll come back and see if anyone else has a specific they want to talk about. So we've covered a couple of things on what's the when is TMT versus DIA appropriate. So it depends a lot on how much protein you can isolate, as we were just saying, and the type of sample. For example, serum samples work a lot better in DIA than TMT. The other factors are time and cost. So TMT is limited by the availability of the different reagents to form the multiplex, and those reagents are expensive. So generally the break point is TMT works well up to about 20 samples, and beyond that it's just prohibitively expensive. And DIA, you can that's almost the sky's the limit there. We can go up to hundreds of samples, and probably beyond once we scale up our platforms a little more. So we could go into some specifics on sample prep here. So cell culture, that's the probably the easiest type of experiment to do, because it's it's easy to harvest the cells in a clean way, pellet them, freeze them, and ship them. That's no problem at all. In most cases, it's easy to get 1 to 5 million cells that we need for any type of experiment. So that's no problem. Tissue samples can be a little trickier depending on how difficult it is to lyse the tissue and how much of the tissue there is. So we want 10 to 100 milligrams of fresh frozen tissue to generate 10 to 100 micrograms of protein per sample in that range. But most of the time it's not difficult to, if you have experience lysing your particular tissue, you can just prepare a RIPA buffer lysate, and that will be perfectly good for proteomics analysis. The laser capture from FFPE tissue from FFPE samples works remarkably well. They're pretty good protocols for extracting protein from those samples. If you can get a good LCM in the first place, that's probably the trickiest part, depending on your institution, the availability of the equipment, and someone who's expert enough to do it properly. If you've got that, then it's definitely a workable experiment. Serum is pretty easy to work with because it's uniform in terms of protein composition. The main the main complication there is consistent handling sometimes. So a lot of the way in which serum samples, especially from patients, have to be collected sometimes works against, at the moment of collection, the future research is not always the top priority. So you can, where you run into trouble is differences in how samples are handled and collected. But if you have a consistent protocol, that's usually not a problem. We don't need much serum for proteomics analysis. 10 to 25 microliters per sample is plenty. Then the last example here is pull down type experiments. And those are some of the trickiest to figure out how to work with. And usually the main complications there are, number one is yield. It's, you can hardly ever get as much protein as you want with a pull down experiment. And the other is detergent. Usually you have to lyse something before you do the pull down, and that lysis has to happen in a context that isn't necessarily LCMS friendly. So typically with IPs, we want to talk to people interested in doing these types of experiments and just take a look at the pull down protocol and make sure it's compatible with our protocols. And there's usually a way to get there, but it's important to have that conversation before the pull down takes place, because if you end up with samples with 10% Triton X100, there's not a whole lot we can do with those. Okay, so on the transitioning to the detergent issue, this is Triton X100. That's the bane of LCMS spectrometrists, and you can see from looking at these figures why that is. So if you have too much detergent in a sample, you get a PEG signal on the chromatogram. You get bad, poor chromatography of peptides, and it's basically the same thing you see on this gel here. Is this gel looks terrible for the same reason that this chromatogram looks terrible. So to the extent we can, and the prep protocols we've got are a lot more forgiving now. We can, minimal, fairly reasonable amounts of detergent can be washed out now in a prep. But we want to, if these substances are involved in the sample prep, we want to know about that ahead of time so we can deal with it appropriately or modify the prep protocol if we need to. And we've already talked about fractionation and varying that to fit the experimental needs, and we've kind of talked about this here. And what else do we have? How much instrument time that's related to fractionation? So this is probably a good time. Well, I'll just talk about these proteomic depth here, and then we can see if anyone else wants to talk about their experiments. So if you can, how much instrument time do you need in an hour? On the newest mass spectrometers, you can ID, 2,500 to 3,000 proteins. In 12 hours, you can get up to, 5,000 to 7,000 proteins. 24 to 36 hours, you can get to 8,000 or 10,000 plus. So it's kind of diminishing returns. So you don't, and this tends to get very expensive if you go out this far. So you don't want to shoot for 10,000 proteins unless you need 10,000 proteins. If if you're looking at a transcription factor, probably so. Otherwise, no, you just want to shoot for the level of proteomic depth that you need, assuming that you're able to calibrate that up front. So does anyone else have anything they'd like to ask about or bring up here?